Hello, here is a video to talk about PhysioEX. We're just going to start with carbohydrate digestion, um, and I'll give you like a brief overview of how it works in case you're still confused about any part of it. Basically this is starch is our substrate, our enzyme is amylase, and our product is maltose, but we are just going to say sugar. So we are tracking the digestion of starch by amylase and looking for the production of sugar. I just want a different color on my pen here. All right. Moving right along then, what else do we care about? Let's just go to the table. Clearly I have not rehearsed this video. So here is my data table. You might have your lab report. And yes, this might be a little bit different than the actual experiment, but it's got all the stuff that I care about in it. So first up is the sodium potassium iodide test. You want to remember for that it is brown in the presence of, or I should say it starts off brown. In the presence of starch, it is going to turn blue-black. That's supposed to be a B. So this is actually a test for substrate, to see if there's substrate left over. Benedict's is a test for sugar. That's a test for our product. And a positive reaction to sugar is yellow to red. I'm just going to say red. Now all I'm going to do when I go over this is, like, if there's no sugar, it'll be that. If there's a little bit of sugar, it's that. If there's like a little bit more, it'll be that. And if there's a ton, it'll be that. So we'll just do different amounts, none, some, a little more, and the same thing with starch. So negative reaction, some starch, some more starch, a whole lot of starch, something like that. So then, let's just go over the different tubes. Tube 1, we have amylase and starch plus a pH 7 buffer. But we boil it first. So what boiling does is denature the enzyme. It's like boiling an egg, right? The egg white turns all hard and everything is denatured, so it no longer works. There was starch to begin with. We didn't digest any because our amylase got killed. So all the starch should still be there. And then because our amylase got denatured, there should be no sugar. Here we have amylase starch, pH of 7. We are supposed to look at this and say these are ideal conditions because amylase is in our saliva and amylase is in our small intestine, which I do not accentuate but does have a relatively neutral pH. The secretions in the duodenum neutralize most of the stomach acid. So here at this pH, 37 degrees, amylase should work. So you should have no starch left over because the amylase digested it all, and then lots of sugar because, again, all the amylase got digested. Here, anytime you see water, this is a control. You put this tube together intentionally with something missing to prove to yourself that all of those things are required to get whatever reaction you're looking for down here. Mostly we're looking for the presence of sugar. So when these tubes don't work, the other tubes that do work make us confident. So we'll go over it better when we get done with this. Sorry. Okay, so this tube right here, amylase, water, pH of 7. So you have enzyme, but you have no substrate. So because we didn't even add any starch, there isn't any starch left over. And because there wasn't any starch to begin with, there's no sugar because the sugar comes from the starch. And then here we see we add starch but no enzyme. So because there's no enzyme to digest our starch, all the starch will still be there and we won't get any sugar. So taken together, these two negatives here mean that you have to have starch and you have to have enzyme in order to get sugar. And sugar is what we really care about because this is the product of our enzymatic reaction. So then looking at this here, right, we know that all that sugar is there because the enzyme digested the starch and not because somebody spiked our pH 7 buffer with sugar accidentally or something like that. So those are called negative controls. They test negative for what you want so that when you do get what you want, you know why you got it. You can trust your positive results. They are not false positives. This one we don't care about maltose. So here we are. We've got enzyme, substrate, pH of 2. So this is 5 whole pH units below the pH at which amylase is usually functioning. So this is not going to be good for the amylase. I would say, if I remember how this works out, most of the starch is left over. And you only get a very little bit of sugar production because most of the amylase is denatured, or all of it is denatured to the point where it's not very functional. So you haven't completely killed it, but it's not doing very well. So same thing here, you have amylase and starch, but the pH is not necessarily where you want it to be, but it's only 2 pH units out of amylase's comfort zone. So it's not as bad. You'll have some starch left over, you'll produce some sugar, but not as much sugar as you get at a pH of 7. And that's how you do that one. Then moving right along, remember we're not doing cellulose. Then we get to this one here. So first of all, our enzyme is pepsin, and our substrate is BAPNA, which is, it's not an artificial protein, but we can call it like a protein substitute. And I forget what it says up there. We're going to say BAPNA is mostly clear. Yes, I just looked it up. It's clear. So it starts out clear. When pepsin digests the BAPNA, or does whatever it does to the BAPNA, I don't understand exactly what this chemical reaction is. The product is, I think it's yellow, but it doesn't matter. It's some sort of color. Let's go up here and see if it says yellow. There, I was right. So yellow is darker than clear. All this machine down here is measuring, do they even show it? I guess they don't, is optical density, which is just how dark is the solution. If something is optically dense, that means light is not passing through it. So the darker yellow the solution becomes, that means more BAPNA was processed by the pepsin. So really our product is color, or yellow BAPNA as opposed to clear BAPNA. And again, we've got different pHs that we're going to look at. And we don't have a test for undigested BAPNA here. It's either BAPNA got digested or it didn't, and we're tracking the appearance of the yellow color to measure that. So again, if we look here, right, we're just going to go no yellow, little more yellow, lots of yellow, instead of writing down what the actual optical density was. All right, then again, so we look at tube number 1. Pepsin, BAPNA, pH of 2. Hey, pH 2, should that work? Should that not work? Let's think about where pepsin usually functions. pepsin usually functions in the stomach. You're all thinking stomach, I know that. And the normal pH in the stomach is somewhere around a pH of 2. So this is actually a good pH for pepsin. However, we have boiled it for however long, a minute maybe, before incubating at 37 degrees. This, by the way, is body temperature, human body temperature in Celsius, 37 degrees. So that's a good temperature for all human enzymes. So we got pepsin, BAPNA, pH 2. It should stay colorless, which means no optical density, because we destroyed or denatured our BAPNA, so it's not going to chew up, we denatured our pepsin, so it's not going to chew up any of the BAPNA. Tube 2, pepsin, so we've got enzyme, substrate, the proper pH for our enzyme, because it functions in the stomach. This we would predict, and you should have gotten, will have lots of yellow color, lots of BAPNA processed into yellow BAPNA by the enzymatic activity of pepsin. Now then we go here, oh, something's missing, we've got water. So it's enzyme, no substrate, pH of 2. If there's no BAPNA, BAPNA is what turns yellow, there will be no yellow. Here you have BAPNA, but no enzyme. If it is true that pepsin is responsible for converting BAPNA into yellow stuff, and BAPNA just doesn't turn yellow when you expose it to a pH of 2, this should also test negative. So these two, again, are negative controls. They are designed to test negative so that when you get a positive result, you can trust it. Again, these two taken together mean you have to have water and BAPNA together at the proper pH in order to get your enzymatic reaction. So then we go here, pepsin, BAPNA, pH of 7, so we've got enzyme and substrate. Our pH is higher than we would like it to be. So let's say you get some color, and then here, your pH 9, you're a little even farther outside pepsin's preferred range, so you've got maybe just a little bit of color. So that's what you should get for this experiment or this activity here. All right, moving right along, this is going fast. Now we're looking at fat digestion by pancreatic lipase with the assistance of some bile salts. So our enzyme is lipase, oil is our substrate, and then we'll talk about what our product is going to be. Bile salts, you remember, just emulsifies, which means it allows the water, and the lipase is in a watery or aqueous solution, to mix with the oil. Now, if they don't mix, then you just have that one line of interface, right, where it's oil on top, water on the bottom. Not a lot of contact between the two, but if you have a whole bunch of oil droplets suspended in a watery solution, then there's a lot more surface area for your enzyme to act upon. Let me just delete those so it doesn't get too busy. All right, then, so to understand how this test works, you have to understand how lipids are digested and what a lipid is. So a lipid we sometimes draw like this. I should make these lines longer, right? So it is a glycerol head group. I don't have room to spell glycerol. With these 3 hydrocarbon tails, sometimes referred to as fatty acids. And I don't know enough about the chemistry of this digestion to say where the hydrogen ion gets released to cause these to be fatty acids, but you should just know when you separate your glycerol from your fatty acids, right, these are going to lower the pH of a solution because that's why they're called acids. So when you digest lipids, the pH of your digestive juice should go down, and that's what we're measuring down here is pH. So we're looking for a drop in pH as an indication that lipids, oil, has been digested, releasing free fatty acids. And I think, again, I'm just going to do a no drop in pH, some drop in pH, more drop in pH, big drop in pH. pH is going lower, but I don't want to do like less pH, lot less pH, less pH. That just looks wrong. So we'll continue with the same symbols that we're using to indicate how much enzymatic activity we're getting, or how much product we're getting that is indicative of enzymatic activity. Ooh, I just said a big word, indicative. Sorry. It's been a long semester. I'm getting a little punchy. So then, tube number 1. We've got lipase. Remember, that's our enzyme. We've got our substrate. We've got our bile salts. And we're at a pH of 7. Lipase, you'll remember, is like amylase. It works in the saliva. It works in the small intestine. So probably happy at a pH of 7. So we should see drop, drop, drop in pH, indicating a large amount of fatty acids being freed from the glycerol, or freed from the larger structure of the triglyceride that they're contained within. Tube number 2, I see water, so something's missing. You've got your enzyme, you've got your substrate, you're at the proper pH, but you are missing your bile salts. And Physio-EX will tell you nothing happens there. I kind of find that hard to believe. There should be some digestion. It's just not nearly as efficient. But whatever, this is what Physio-EX, I think, spits out. And again, it's because without the bile salts, the watery lipase solution does not mix with the oil, and so there isn't as much or enough contact between the enzyme and the substrate. It just can't get at it very well. Then you have here, let's see, you've got enzyme, you're missing your substrate. This is clearly a negative control. They went and changed the pH on us for some reason. I don't know why the heck they did that. But you're missing your substrate, so you're not going to get a drop in pH. And here, there's water. You're missing your enzyme, so you're not going to get a drop in pH. Here, you've got everything you need, enzyme, substrate, bile salts, but your pH is 2. So you are not going to see a change in pH, really for 2 reasons. One, the pH is already 2. It's hard to get lower than that. And 2, at a pH of 2, lipase is going to be exceedingly unhappy. So there's going to be very little enzyme activity anyways. Then here, you've got enzyme, you've got substrate, you've got bile salts. Your pH is off a little bit, but you should see some drop in the pH because at a pH of 9, the lipase in this case still should have some activity. And I will point out, I'm not entirely certain why, but I think I know why. Most of the time, an acid, a drop in pH will alter the enzyme's activity more than a rise in pH. So if this was like 5 versus 9, so 2 pH units lower versus 2 pH units higher, you'd probably see more activity higher than lower because acids are just better at denaturing proteins than bases. And I believe it is the actual presence of the hydrogen ions that interact with the protein, whereas with a base, it's an absence of hydrogen ions, or just less of them, and that doesn't seem to have as much of an effect. Let's see. And then that's it. So for all of those, know what your enzyme is, know what your substrate is, know how the tests work, and then for any tube, be able to say whether or not, or what kind of result you're going to get, and why, and be able to identify your negative controls. That's it. 20 minutes, piece of cake. Thanks for listening this long.